Hi, I'm Lawrence Wong. I'm the founder of Thermagenix and a professor here at Brandeis. We are a company dedicated to a reaction called PCR. And we got started in 2016 with a portfolio of technologies developed in my lab over the last 20 years. And then we've moved out to Natick, in a nice space in Natick, with an initial startup financing, and we are actually looking for additional financing. I've been working with these three guys for about 20 years, and then Charles, who's here, joined us, and Peter Kozin, who's unfortunately in Hawaii. So Thermagenix is dedicated to upgrading PCR. What is PCR? The polymerase chain reaction. Polymerase chain reaction is a process for taking a small amount of DNA and growing it up into billions of molecules in a single tube. It's critical technology for molecular biology research, for diagnostics, and for next generation sequencing. All of you are sort of intimately aware of next generation sequencing, or will hear a lot about it because everybody's genome is going to be tweaked to figure out what you might or might not be coming down with next week. The problem is PCR makes mistakes, and in making mistakes can give unclear or incorrect answers, and so there's a really critical need for improving PCR, and that's what we do. We've invented 3 different types of molecules, which are called Thermostat, Thermago, and Thermostat Party, which very simply, we just add them to reactions and they clean up the mess. And there's some illustrations of that over in our show over there. So our intention is to make PCR error-free. Now, to give you some idea of the market for this, the total market for PCR last year was $7 billion. Of that, the enzymes involved are critical, and a reagent called Hot Start, which comes in various forms, only partially works. And yet, it adds about 50% of the cost of the enzyme. Now, Thermostat, Thermago, and Thermostat Party are much better than Hot Start, and not more expensive, for sure. And therefore, if we just capture 10% of that market, of the PCR market, we would be doing well. We estimate that our serviceable available market would be 150 million to 250 million per year. So that's kind of motivating. We sell Thermostat, Thermago, and Thermostat Party directly. So we send out email blasts, 40,000 blasts at a pop, and our sales are taking off. We've really only been up and running since September, and that's very nice. We also have global partners, distributors. One is Millipore Sigma, who have a non-exclusive rights to sell our reagents globally. Funakoshi in Japan, and Cambridge Biosciences in the UK, and we expect to add other distributors to that as well. So we're in a mode of advertising and getting the word out there. The reagents work very nicely. So we anticipate substantial growth in 2018. I'm done. I'll take questions.
Take questions.
Yeah.
Can you briefly explain the reason of adding those couple ingredients to make it error-free?
Okay. So for the last 20 years, people have tried to make PCR error-free by adjusting the way in which DNA strands interact. We hit upon these set of reagents which actually interact with the polymerase, the enzyme that does the work, and alters the specificity of the polymerase. It makes the polymerase choosier. So the polymerase only does the right thing and doesn't do the wrong thing. So it's fundamentally different property to how to cure the problems with PCR.
So in that regard, then it kind of impact the replication process, the speed of it, to make the reaction much longer, is it?
No, it doesn't slow down the reaction. It causes, in fact, what it does is the enzyme rejects the mistakes and chooses the right, what we call primers, the perfectly fit primers. So we don't inhibit the reaction, we enhance it. Anybody else? Yeah.
What does the RT stand for in your program?
Reverse transcriptase. So the process, when you want to study an RNA, which is the kind of molecule that takes the information in the cell nucleus and moves it out to the cytoplasm, that's an RNA. The typical way to analyze that is to convert that RNA back into DNA. That's called reverse transcription. Those enzymes, which are different from polymerases, are notorious for making mistakes. And so there's a whole industry around making those enzymes convert RNA into DNA, and we think that virtually everything out there is junk. If you look at the illustrations over there, you will see that it makes an awful lot of mistakes. We take Thermostat Party and we add it to the reaction, and amazingly, it makes just the right stuff.
Great, thank you.